So thanks for joining today. This is a course that we are calling CFDE Power Up. And so I'm very excited. We've offered a couple different training programs before, and so this is the first time we're offering this particular one. So a couple kind of housekeeping notes at the very beginning is that I'm sure Zoom probably informed you that we're recording the session today. We will try to make the recordings available to the course participants, and then we may cut out parts of the recording later into like kind of smaller videos. It's largely going to be the instructors and the websites that we're sharing, and so just a heads up on that we are going to be recording. All right. So like I said, this is the first time we've offered CFDE Power Up before. So let's talk a little bit about it and what we're going to do. So we have two instructors for the course. My name is David Higgins. I work at the Children's Hospital of Philadelphia, where I'm the informatics program manager. And I work on especially closely related with a project called the Kids First Data Resource Center, which I'm going to talk about a little bit later. So my co-instructor for the course is Sangeeta, and so I'll pass it to her and let her say a couple words about herself and introduce herself.
Thank you, David. Hello, everybody. My name is Sangeeta Shukla. You can read from the screen. You can look at me. I'm Sangeeta Shukla, and I work as a bioinformatics scientist for the Children's Hospital of Philadelphia. And over the years, I've worked actively with a lot of bioinformatics pipelines, mostly custom developing scripts. And eventually, I found out about this wonderful cloud workspace called Kavatica. And today and the not today, but during the upcoming days, I am here to mostly just share with you all the things that I've enjoyed learning about using bioinformatics data on that Kavatica platform. And I'm looking forward to making sure that I share with you all as much as I know, at least, and hoping that you all enjoy it as much as I do, at least.
Great. So we've got two, like I said, two instructors today. So the main structure of the course is that Sangeeta and I are going to alternate days. So I'm going to be running today's session, and then Sangeeta's going to be running on Tuesday, and then I'll pick it back up on Wednesday. But the other person is still going to be around and can try to help answer questions in the chat and things like that. All right, so that is that. So here we are on day one of the workshop. And so I just want to provide an introduction to what we're all about and what we're going to cover today, as well as later this week. And it's really going to be kind of an introduction, orientation, kind of exploration kind of day, as I'm going to talk about once we get into the content of the course here. So we're going to learn a little bit about what the CFDE is and what these different DCCs are. We're going to look at the types of data sets that are available and kind of start to explore how you could access data for some of these projects. All right. So what is our goal? So introduction to the workshop. So why are we here? So our goal for this workshop is to teach everyone skills in using common fund data sets to support your research in pediatric biology. And so this is kind of a very, very lofty goal and very broad goal, but I want to highlight one word. And so specifically, we want to teach you skills in common fund data sets to support research. And so from our intro here is in the chat, it sounds like we have folks from all over the country, which is great, investigating lots of different topics and different conditions. So we want to try to reach everyone individually. And so we asked you a couple different questions in your pre-event survey that you filled out. And so one of them was this question: how do you use omics level data as part of your research? And so we had a kind of a range of, kind of choose your own, self-identify which category you fall in. And we had a really broad range of folks. And so some people, responded that they're very comfortable with genomic techniques. They do them, perhaps daily as part of their work. All the way down at the bottom, where some folks, identify as, well, I don't have a lot of skills at all. I'm hoping to learn more in this workshop. So as the instructors of Sangeeta and I are trying to put this course together, we're really trying to think about, well, how can we, to go back to our mission there, how do we teach you skills when you, represent a very broad collection of skills and expertise that you bring with you. And so the way that we've decided to structure the course is we have set it up over the next 5 days to kind of scale and go from kind of very basic to more complex, leading to Friday, which is where we're going to explore the next steps. And so on Monday, that's today, we're going to have, like I said, a very kind of introductory day. We're going to talk about what CFDE is. We're going to explore this concept of these DCCs, and we're going to look at some different sites and learn about how to access data. Tomorrow, we're going to dive. So once we kind of get a sense of what is out there, then we're going to explore, well, how do we access these data sets? Excuse me, how do we use this data as part of an analysis? And so we're going to look at different tools and methods in bioinformatics in terms of different computational tooling, as well as different experimental design techniques. And then Wednesday and Thursday, we're going to actually do some analyses together. So on Wednesday, we are set up to look up how to do an RNA-seq analysis using a software tool called TE-seq2 in Cavatica. We're going to be looking at some kids first data, probably some RNA expression data, probably from leukocytes from the kids first data set. And we're going to be looking at comparing gene expression. And then on Thursday, Sangeeta is going to lead us in an analysis where she's going to be building a single cell RNA-seq pipeline in Cavatica. And I believe she's using some expression data from HubMap as part of her demonstration for that. And then Friday is kind of the like, we've completed the course and now we're moving on to where can we go next? What can we explore? How can you take what you learn here with you? So the design of the course is meant to kind of, as you see, scale. We're starting on Monday and we only have a week, so we have to scale very, very rapidly. But the goal is that a couple goals here. One is that each of these days is independent of the other days. And so if you need to miss a day, if you need to, if you're perhaps are already, feel like you know information about a day, that's okay. Hopefully each day should be self-contained on its own. And so you can get that content by itself. But also, the days kind of scale. And so we start from the beginning and we get more and more complex as we move on. So if you are, as I said, so to go back to our previous, if you're a respondent, one of those 7 folks at the very top who says they're very comfortable with computational techniques, maybe Wednesday and Thursday might be better for you because we're going into, specific details of how we're using these data sets. And whereas if you're, one of those 5 respondents who says they don't have a lot of skills in computational biology, maybe Monday and Tuesday might be better for you. Wednesday and Thursday could be very advanced to try to jump from Tuesday to Wednesday if you don't have a lot of skills. But we can, on Friday or on late on Tuesday, we can provide some opportunities, different resources that we have found useful in terms of learning some of the basic skills in order to be able, so that way everybody can kind of take what they learn and move forward. And so that kind of brings me to my last point about the intro here, is that we're really trying with this course to kind of lay out a very broad overview of a lot of different topics. And so we want to kind of introduce participants in the course to a lot of different things. So that way we can share with you, this is out there, these types of things exist. In our span of our week, we don't have, the opportunity to be able to like drill down deep into all of those different topics, but we want to share what is out there and then hopefully encourage folks who are interested in, oh, well, I learned about this, so I want to, learn about this more on my own independently. So that's kind of our goal here of the course, is to kind of introduce a lot of different topics and then encourage folks, to follow up on Friday and then afterward with how do we kind of learn more about that independently. All right. So that is my introduction to our workshop. Okay. So today, and I see some folks sharing in the chat about the types of things they are interested in learning about, which can be great. So right now, especially on Friday, we have some opening in our agenda. And so if folks have specific topics that they're interested in learning about or hearing more about, yeah, please put fill up the chat, and we'd love to read it, and we can continue to develop the course as we go on our way all the way to Friday. All right. So today, to go back to my agenda here, so we're going to introduce the Common Fund data ecosystem and this concept of these organizations called DCCs. So before we can get to the Common Fund data ecosystem, we have to kind of back up a level. And so we need to learn, well, what is the NIH Common Fund? And so the Common Fund is a program in NIH. NIH is the National Institutes of Health. And so institutes is the key word, plural there, because there's lots of multiple centers that are all part of this big umbrella of what's called the NIH. And so within NIH, there are different centers that are all focused on different conditions. So there's like a heart, lung, and blood center. There's the NCI, the National Cancer Institute. There's a pediatrics center. There's a kidney health center. And so there are lots of different centers that are all kind of focused on a particular aspect of human health. So what the NIH Common Fund is kind of this interdisciplinary program that exists outside of each of those individual centers. And it supports these collaborative interdisciplinary projects where multiple institutes might be working together on research that's supposed to make a broad impact. And so the Common Fund has a couple different programs that are all kind of in its organization that we can talk about. So what are some examples of these programs that exist in this Common Fund structure where we're supporting, perhaps, not just kidney health or not just cardiac conditions, but kind of very broad data sets, broad projects that could be used in multiple different contexts in terms of medical research. And so I'm going to be jumping back and forth between slides and a slideshow here. So I'm going to be sharing my screen. So the first one I'm going to talk about is this program called GTEx, GTEx. So this is the Genotype Tissue Expression Program. So what this is a program where they've been investigating the relationship of genomic variants and gene expression. And so it's this really interesting data set. It's a catalog of samples, 54 unique tissues from 1,000 deceased participants. And so it's very big in terms of the number of participants, but then also it's very interesting because it is a collection, as we said, 54 different tissues from the human body. So from your head to your toe, there's, depending on what you're researching, there's probably a sample that could be useful to you as an investigator. So I'm going to stop sharing here, and we're going to switch over, and I'm going to share my screen. Actually, let me back up here. So I think I'm going to go back to my slides, and then we're going to share all at once. So we'll do GTEx first. Okay, so we're going to share this button, and we're going to click this button, and we're going to click this button. Okay. So this website is the GTEx portal. Where's my chat? There we go. And so this website is a kind of the portal site of how you would be able to access the data for this program. And so they have a couple different kind of iterations of GTEx. So kind of the classic GTEx that was published and is the project that's complete is available here, but they actually have new projects that have been funded that aren't available yet, including across different developmental stages, as well as comparing human expression to non-human primate. So some additional data sets there. So the GTEx portal offers data in a couple different formats. So for example, as far as the data that they have a lot of different open access data sets. And so they have, as I said, they have very interested in gene expression. So they have a lot of RNA data. So especially if you're doing an experiment where you want to compare the expression of, you're perhaps a tissue, maybe a tumor tissue or a tissue with your condition to a control tissue, you can use the RNA data from GTEx for that. They also have some QTL data in terms of association, how do you associate that expression, as well as information about the donors themselves. And so what are the types of people that they have expression on. And yes, there is some long read data. It is available. And so we're starting to move into the long reads technologies. The GTEx long read data is up. And I know some other programs are starting to put their data as well. And GTEx also supports protected access data as well. And so, if you're interested in, the raw files, the alignment files or the VCFs for this whole genome, the called variant files, these files are available, they are protected access. And so you would need to apply for data access to be able to use those. And so it allows users to be able to download specific files, which we'll explore a little bit further on Wednesday when we look at our expression analysis. And so you can sort based on, particular tissue. But you can also, so it's a portal, so it's more than just download the data or copy the data, is that you can also explore particular genes as well. So for example, we can type maybe our favorite gene is BRAF. And so it pulls up, it gives you information about what BRAF is, and then here's those 46 different tissues, and it tells you roughly, which tissues throughout the body it's particularly expressed in. And so I won't, kind of belabor the point, but it gives lots of different information, about different expression tools, as well as if you're interested in, so those different tissue types. So we said there's 46 different tissues. So it kind of explains, well, what are those and how do you define those and how are those samples collected? And so if you're interested in in the colon or in the bladder, you can find specific tools. Tim is asking, we used to pull raw data from dbGaP. Is it feasible to interop on Anvil access across CFDE data sets? So yes, it is getting easier. And so that is part of what we're going to be able to show is how we can move data between different platforms. And so that's what we'll show as part of our Kids First demo, and the HubMap demo is kind of moving data into Cavatica, which can then, also different tools work similarly. Yes, saves about 100 terabytes of storage. Yes, exactly. So, as I'm going to talk about, a big part of this tooling is that we make this data available in the cloud, so that way folks don't have to copy and have their own copy. If we can all access that same copy that's up in the cloud, then we don't need to do as much copy pasting, which saves space, which makes everyone happy. Okay, so I'm going to switch back to my slides really quickly. I did not think about this when my slides were in their own presentation. Usually I do Google Slides, but that's okay. All right, so I'm going to share my screen again. So that's GTEx. So you can see how it might be something that could support a broad set of research topics for a broad number of researchers. So another Common Fund program that's very familiar to me because I work on this project is the Gabrielle Miller Kids First project. So the goal of Gabrielle Miller Kids First is that we're very interested in pediatric conditions. We're especially interested in the underlying genetic causes of pediatric cancers and structural birth defects. So data at Kids First is a collection of 35 different studies, and each of these studies has genomic data on a collection of pediatric patients that all have a shared clinical condition. So I'm going to back out of here and let's take a look at what this looks like. So I'm going to stop. There's going to be lots of switching while I go back and forth. All right. So go back to here. And so that was GTEx. So my second tab here. So this is Kids First, the Kids First Data Resource Center. And so we have information on the website about the particular data sets. what types of features they are and what types of files are available. We also have information about the particular studies. And so, for example, if you are an investigator who is researching orofacial clefts, we have several different orofacial cleft studies. And I'll allow individuals to kind of scroll through and explore on their own. Let me share the link here in the chat. All right. So then we have a portal. So this is kind of this site is kind of teaches you a little bit about the background, but then if you want to access the data, you can log into the portal. And so there's those 35 studies with more than 28,000 participants. So you log in. There's a couple different methods to log in, but if you have an NIH RAS ERA Commons ID, you can log in with that. If you don't, that's okay. Any Google authenticated email address, you can log in with that as well. So I'm going to use my NIH authorization. And so this is inside the portal now, so you can explore in a little bit more depth in terms of, so here's that list of 35 studies, as well as the types of data that's available in terms of either genomic data or transcriptomic data, things like that. And so you can explore. So here's these studies, and so they're these very broad categories, but you can explore in a lot more depth by going into the data exploration tool, where you can actually query these different data sets. And so you can search across particular studies. So if you're, really only interested in, maybe the leukemia study, then you could select, just that particular study. And it will tell you information about the participants in the T-cell ALL. Or you can search based on condition. And so, for example, I know that a couple of folks said that they're interested in craniofacial or facial clefts. So you can explore under the clinical tab, you can search based on phenotype. And then you could type in, so this is an HBO ontology. And so you can search oral cleft. And it turns out that there are 1,500 participants who have either oral cleft or the term underneath, which kind of rolls up into oral cleft. So we can apply that. And so that lets you have a little bit more information, about these folks, what types of study they're in. So a lot of them are in these orofacial cleft studies, but then, if we scroll back to our study names here, we see that there are, so this is the structural birth defects associated with chromosome 18 copy number, and charge syndrome and congenital diaphragmatic hernia, and the Children's Brain Tumor Network. And so there are, participants in these other studies that also have oral clefts, and this kind of gets into the underlying, cross condition investigation of Kids First. So you can search based on the studies, but also search based on the specific conditions. So Kids First, so once you have that set of participants, you can explore the different data files that are available. Kids First has a couple different data files, so they support, alignments if you want to do the kind of very start from scratch beginning, but they also support called variants as well if you want to start from the VCFs and do your own research. So this is Kids First, so it lets you, again, it's really focused on pediatric conditions. And so to go back to my slides in the previous, to bring us back to our Common Fund intro here, we're very interested in kind of broad topics. And so obviously there is, as I said, there is an NIH institute specifically focused on pediatrics. But pediatric conditions, such as heart conditions, affect people of all ages as well. And so it's again another example of a very broad data set with lots of different folks. Could be available and valuable to lots of different researchers. All right. So then Tim is asking what references. So we are using HG38 in Kids First right now for the GVCS. And I can share the pipeline as well if you're interested a little bit later. Let me check on that. Or maybe Sangeeta can check on that. Yes. All right. So then the last one that I'm going to talk about is HubMap. So HubMap is the Human Biomolecular Atlas Program, HUBMAP. And so the folks at HubMap are very interested in developing this open global platform to map healthy cells in the body. And so they're very interested in kind of biology on the cellular level. So we're not just tissues, we're a collection of cells, and how do these cells arrange themselves, talk to each other, and ultimately, form tissues and organs and human beings. So they have a collection of a lot of, since they're very cell oriented, they have a lot of single cell data. They also pair with microscopy imaging of human tissues as well. So for one more time, we're going to pop out of here and we're going to pop into our browser and we're going to take a look at what HubMap looks like. So let me share the screen again. This one. All right, so we're going to say goodbye to the Kids First portal. So this is HubMap. So I will put this in the chat as well as our third demo. This one. That one. So HubMap, as I said, there's their same explanation that they're interested in developing this framework for mapping the human body at the cellular level. And so we can learn more about what types of data they have. They also have their own portal, this HubMap data portal, as well as this human reference atlas. And so we can explore the data portal. Yes, we can open a new tab, that's just fine. And so we have information about, the participants or the donors, the people who are donating the particular tissue to arrive. That appears, in the database. So where were these patients consented and where was these samples collected from? There's also information on the particular samples themselves. So, where is this tissue being collected? And then all the way down to particular data sets. So we could click on a particular participant. So this is a kidney sample. And so we can scroll down and we can learn more about what types of data has been derived off of this kidney sample. And we can see that they have single nucleus RNA data available. And so we could back out. And so if we wanted to select, for example, if we were investigators who were researching the large intestine or the colon, we could select that. It seems like there's a study at Stanford that has lots of different colon samples. We can explore each one in detail. So also in GTEx, so GTEx has a lot of this, single cell, single nucleus data. They also have some, looks like they have some epigenetic data. So they have some ATAC-seq as well. And so we can explore a little bit further. They also have microscopy imaging. And so if I pull up, it's pretty cool. You can actually view the images right in the browser. And so it allows you to be able to pull up and see the particular imaging sample. And I'm not an expert in kidney biology, and I'm not an expert in this particular sample, so I don't know how to explain what we're looking at here. But it allows you to see that you can see, connecting that imaging data connection of what you're, where this sample came from. All right. So with that, so those are my three examples of these common fund programs. But again, with that one, you can see we're looking at lots of different tissue types, kidneys and colons and hearts. And so it is, again, a very broad data set. Okay, so if I share my screen again and click this and click this, okay. So I chose those three particular programs because we're going to explore the data a little bit further throughout the rest of the course. But there are a lot more than just those three, as we'll see here in a couple slides. But that's three examples of these common fund programs. And so by going through that, hopefully it's clear that each of these programs has their own unique goal. their own unique data sets, and they've got their own website and their own platform, and everything is set up such that you can work in that space and carry out research projects that are, working towards that goal associated with a particular platform. But at the same time, you can imagine that, well, how do we combine some of these data together? How could I take the expression data in GTEx and maybe compare it to expression data from tissue in Kids First for tissue that has the particular condition? Or, how can I combine GTEx data with HubMap data, which also has, single cell level, whereas GTEx has whole tissue level. So kind of combining these things together. So as you can see that, each of these programs is their own independent platform, but we could really benefit investigators by combining this data together. So data from each of these projects could help inform projects where we're combining and getting these larger research questions, better research outcomes, and better discoveries. So how do we, if there was only some type of way that these common fund data groups could be coordinated together? And so there is the Common Fund Data Ecosystem. So the CFDE, GTEx system, single nucleus data. Anyone done a demo? I have not done a demo. I know that Sangeeta is doing a demo with single nucleus, but I'm not sure if she's going to be pulling in the GTEx data with that one. So we have CFDE, we've done it. So what is CFDE? So CFDE is the Common Fund Data Ecosystem. And so the goal of the CFDE is to do exactly that, to be, those arrows that are moving around there, helping investigators be able to kind of combine data from different of these independent sites and help combine it together in a way that's findable, accessible, interoperable, and usable by participants, by researchers, and reusable. And so the way the CFDE works, so here's another graphic from the CFDE site. And so there's CFDE sitting right in the middle. But then all around it are these different individual ICCs or DCCs. And so here's the three that I highlighted: Kids First, GTEx, and HubMap. But you can see there's a very broad collection of different sites out there. And so, I didn't get to talk about, the metabolomics group or the 4D nucleome group, which is all about, genomic structure. There's also, what else? There's also MotorPack, which is all about muscle biology. And so there's lots of different groups that are, each have their own particular focus that allows, we want to be able to try to support combining this data together. So each of these programs, each of these little squares on the outside is its own what's called Data Coordinating Center, or DCC. And so each DCC is kind of independent in that they're responsible for managing their own project, their own data products, and making that available to investigators as they might want to use it. So what the CFDE does is it serves as this coordinating center here in the middle to help coordinate all these different DCCs to make their data products more easy to recombine together. And so that's what we're going to be, showing here in our workshop on Wednesday and Thursday. All right. So CFDE also has a site, and they also have a portal. So let's go check that out for today. So let me go here. I'm going to be really good at sharing my screen here by the end of this. We're going to stop, and then we're going to click this, and then we're going to click this. Okay. So CFDE has a couple different sites that they support. So this is actually their older site, their search portal. As it says up at the top, they are going to be discontinued this summer, but I wanted to share this one, and then we can explore their newer version as well. So let me pull up the chat and post this here like that. And so So the Common Fund Data Ecosystem Search Portal. So what this tool does is it lets you explore what types of data are out there, and what types of, across all of these different DCCs. So that around this web, how can we search all of these things at once? I don't want to go to the Kids First site and then go to the GTEx site and then go to the metabolomics site and then go to the glycoscience site and do like 12 different searches and then try to piece it all together. So that's the goal of the search portal and the new workbench, is that it allows folks to be able to search one place, and then it kind of searches all of those little rectangles for you, and then it says, oh, Kids First and Motor Pack and metabolomics have this type of data. So it lets you kind of search in one spot, which is super helpful. So you can browse the different types of data, or you can search. So for example, if we're interested in a particular analysis, maybe we're interested in RNA sequencing, and so it's sorted based on this ontology code. Let's see here. And so I can pull up, as I can see this list of particular different ontologies. Let's see here. So if I clear my filter and go back to the dashboard and then go into, for example, data browser, let's look at maybe the files. So there's 11 million, almost 12 million records in here of different data files, but it allows you to be able to kind of narrow that down. And so, depending on a lot of different formats. And so you might start with, well, I'm a biologist who investigates, perhaps you could type maybe kidney. And there's lots of anatomy parts here. So we could say maybe kidney. And we can learn about what types of data is available for the kidney. Wow, there's 123,000 different data files where the samples were collected from the kidney. So it gives us information, well, what types of data is in here. And so this is data from Links, which is the network-based, it's very interested in cell networks. They have some proteomics data. If we scroll down, we have lots of links in here, but we could also select based on our Common Fund program. And we have lots of links, but we, information, let's see here if we choose, nope, okay. Show more. All right. So we can explore the different data sets. We can also explore data types. So if we pull up like file formats, so maybe we're interested in, particular alignment files. Maybe we're interested in genomics. And so we can pull up file format. And we can choose, we can see that we've got some alignments. Maybe we choose BAMS as we search, and so we can see that we have lots of different, alignment. And so here we've got the Human Microbiome Project, and they have some virome sequencing assays in their particular samples. So you can explore different data sets across the different DCCs. So this is their old site, as I said. It is being decommissioned, and so everything is a little touchy. But if we go to the CFDE workbench, this is their new site that they're in the process of switching everybody over to. And so the CFDE workbench kind of exists in two different structures here, the information portal as well as the data portal. So the information portal gives information about, what is CFDE. So here's each of those independent DCC sites that I was talking about. If we scroll through, there's Kids First, there's GTEx, and there's HubMap, as well as the other ones that also exist for different particular studies. And then you can explore, they have training and outreach. And so here's our event that we're at today, right now. Hello. But they also have, I really wanted to highlight this one. This was not planned, but actually tomorrow afternoon, they're doing a hands-on all about how to use this new tool, the Playbook Workflow Builder. at 1 o'clock. And so this is hosted by the Links group, and they're going to show this Playbook Workflow Builder, which is the new platform that they are very, very excited about here at CFDE. So if you're interested, if you're available tomorrow at 1 o'clock, our course will be over by then, so you will be able to do both if you want a full CFDE day, but definitely check that out. And so here's lots of other training events for folks if you're interested in learning more. All right. So then we also have, so this is information portal. This is more of kind of a, learn more, what is CFDE, but then there's also the data portal where we can explore the different common fund data sets. And so, for example, we can search if we study a particular gene, maybe we study this gene MCF. Then we can MCF7, then we can see, well, what types of data is available. And so we get gene expression data from the Links group that has this particular, let's back up. Let's search for, maybe we search for BRAF. How about that? Our favorite gene. So it gives us information about, what is BRAF. So if we weren't familiar with that, we could click on this and learn about what BRAF is. As well as a set of information. So how do we learn more about it? So we have information about the expression of BRAF in the kidney through GTEx. And we can click through other, multiple different iterations here as well. So I can back out of here and back out one more time. But it also allows you to search, so if we have BRAF, it also tells us these data sets. So for example, Links has genes that are upregulated and downregulated by BRAF. And so we can kind of explore these different gene sets that have been produced by these different groups. And then the way to kind of connect it all is with the Playbook Workflow Builder, which I said they're going to do a great demo of tomorrow afternoon. So I will support and promote that one and allow you to explore that one further. So CFDE Workbench, so now we're in the data portal. And so we have different tools as well in terms of how to combine these different data sets together and where we're working on trying to combine. So for example, we're really interested in how do we make our RNA sequencing data, since many projects have RNA-seq data, how do we make it more interoperable and able to be combined? All right. Well, thanks for stopping by, Tim. It was great to see you. We'll see you tomorrow. And we will kind of continue. So I do have a little bit of wrap up here, but I think we are going to end a little bit early today. So let me just pull back up our slides. And I will kind of close us down for today, and then we'll have an opportunity if you have questions for me or Sangeeta. Otherwise, that's the conclusion of our presentation today. So let me switch my screen one more time, and we're going to share PowerPoint. I just said I'd be so good at this, and now there it is, that one. Okay. All right. So today we talked a lot about what types of data is available and how you can explore these different sites. And so we're especially going to get into the methods for data access, on Wednesday and Thursday, where we're really going to dive in, kind of introduce those portals, those different sites, but how do we get the data and move it into one place? So that is coming a little bit later. And then on tomorrow, we're going to have a great talk by Sangeeta all about tools and methods in bioinformatics. And we're going to learn about different computational techniques, just kind of a broad overview of what is out there and what are these different things. And then Wednesday and Thursday is when we're going to get into the really kind of deeper diving stuff. This is where we're going to be on the platforms doing this analysis, building tools and running workflows and things like that. And then Friday, there's going to be an opportunity to connect, so we'll be able to chat about where to go next. We believe we're going to have somebody from the Valsera Cavatica team who's available to answer questions about, how do you use the platform? How do you get started? How do you get credits to support your application and things like that? So that's what we're going to talk about on Friday. So with that, I think I'm going to bring it down for a landing here at the end of our session today. But hopefully this was a good introduction to the types of things that we're going to be covering good introduction to what is this concept of CFDE and what types of data are out there. And then, yeah, starting tomorrow, we're going to explore what types of different data sets are available. All right. So with that, I'm going to stop sharing, and I'm going to say thanks so much for joining. If you have specific questions, I believe Sangeeta and I have time to hang out and answer questions. But otherwise, we'll see you again at the same time tomorrow, where we're going to learn about these methods in bioinformatics.